Thanks for the invitation, Larry. And as I express appreciation to Larry for inviting me, I should also say he was one of my mentors in my residency training. And it struck me that that you could both be a good pathologist and also be a good scientist. And Larry embodied that and was a role model for me as I continued my training and wanted to do research work related to the science of pathology. And what I'm going to talk to you about today, I have sort of 2 lectures here, but only 45 minutes to give them, so I'm only going to give probably the first one. But before I even start, you can see the first one is called High-Plex Methods for Biomarker Discovery. And so that's what we'll talk about first. These are my disclosures, and it's important that now I think we're okay for CME because I've made my appropriate disclosures. But I think it's more than that. I've been asked, why do you do this? And for one thing, when I serve as a consultant, as I do for all these companies, you learn about what's going to happen next because you learn about confidential things that the companies are doing that you can't disclose, but then you know about. And you also get to advise them. So sometimes, but not always, in fact, most times not, they take your advice. But sometimes but either way, you kind of know what's coming. And the other thing is I need to disclose that much of the work in my lab, because of the translational nature of the work, is actually sponsored by for-profit organizations, not by the NIH. I have NIH funding and BCRF funding and some other funding that I'll cite at the end. But a lot of my work is funded by companies because it's also in there, it's a mutual benefit that if we're going to try to make something translational. So this is what I'm going to talk about. I'm going to talk about measuring protein on slides, which has been really the focus of my career. And then I'll talk about high-plex discovery tools, in particular digital spatial profiling. I'll emphasize that because I'm not going to do a review of all the high-plex discovery tools. There's too many to count. Then I'll talk first about protein and then mRNA, and probably have to stop there. If I go fast, and then I can talk about bringing a quantitative assay to the CLIA lab, which has kind of been my lifetime goal for my career, which is to take something that we do in our research lab that's quantitative and can potentially better serve patients and actually bring it into a CLIA lab. And so I'll talk about that if we have time. First thing I want to talk about is measuring protein on slides. And measuring protein on slides, there's sort of 2 broad compartments in the way you can measure protein on slides. And when I say measuring, I want to distinguish that from reading. When a pathologist looks at an IHC slide, they read it. That means they give a subjective impression of what's on the slide. When you measure a slide, it's objective. And measuring allows you, there's a couple different ways to measure. The most popular way is to measure the way pathologists measure, that is, count the cells and say what percentage of cells are positive or some sort of parameter related to cell counts. And that's what pathologists do. And your measurement is the percentage of positive cells over the total cells. And this is how we do PD-L1 and how we do a lot of Ki-67. A lot of pathology quantitative parameters are not really quantitative, they're red. And they're not truly quantitative, but they're quantitative light because they are quantified, but they're not necessarily highly reproducible, which is what I would qualify for quantitative. If you want to measure cells, that's fine. If you want to measure tissue, I like to think of it as what we do is measure by molecular compartmentalization. And I use this term, and I'm trying to popularize this term, although most people don't know what it means. What it means, in contrast to segmentation, is that you define the regions on the slide by a molecular interaction, mostly an antibody staining and definition of a mask or a molecularly defined compartment, as opposed to cells. When I say segmentation, that's when you count cells, and then you can have the number of positive cells that are above a threshold for a marker divided by the total numbers of cells that you counted, because you counted the cells. When you measure tissue, you're measuring within a molecularly defined mask so that you cover the whole surface, for one thing, and the second thing is that some of what you cover may not be cellular. And that, I would argue, may be even more interesting than the information you get from the cells themselves. So here's an example of an immunofluorescent image. And then if you molecular compartmentalize that image, you can see the molecular compartmentalization here. And this is NanoString's approach to molecular compartmentalization. Much to my chagrin, they don't call it molecular compartmentalization, they call it segmentation, which confuses it with what the terminologies that are used for segmentation of cells. Here's segmentation. This is bright field segmentation done by QuPath, and you might not be able to see it so well, but you can see each cell down in this field has a nucleus, and then around it is a certain area, and then a line, and each one of those is thus defined as a cell. And you can do that with immunofluorescence as well, and segment, and then you can count. And I don't have an image of how, on the lower corner here, you can see a yellow or a green or a red dot in every cell. And so you can count the number of cells that are quote unquote positive or above the threshold for that biomarker. So in that way, by thresholding and counting, you imitate what a pathologist can do. I would argue if you do that, that's great, because then you've replicated what the pathologist could do. I could also argue that that's not so great, because then you've limited yourself to only being able to do what a pathologist can do. And we hope with a machine we can go much further than we can do by eye as a simple pathologist interpretation. There is one machine that actually gets this right. So the problem with segmentation is it only gets it right about 80 or 90% of the time. And not only that, but if the nucleus isn't there, it won't find a cell. But that's not always the case. There was recently an approach to looking at segmentation like stars in the sky, and it's called Astropath, and it was popularized. Alex Saleh did this with Janice Taub at Hopkins, and Alex Saleh is the guy who worked on the Hubble Space Telescope. And he then made images, and you can see an example that was published in Science of the way segmentation works if you just use one marker, the nucleus, and the way segmentation works if you use 7 markers. And so I think that there is hope for segmentation, but it will still suffer the limitation of only measuring the cells, not measuring what's not cells, which I think is really interesting. And that's what I'm showing here, is that sort of the advantages and limitations of segmentation versus the advantages and limitations of molecular compartmentalization. And I won't read through these things. You can watch them later if you wish, or read them in various reviews. But remember that if you take away one thing, you remember that segmentation measures cells, and you get positive cells per total cells, and molecular compartmentalization measures tissue, and you can get it attomoles per square millimeter. So you actually get essentially a concentration like you would in a biochemistry lab or in blood or something like that. And so why do I think molecular compartmentalization is better in most cases than cell segmentation? And here's the reason here. Here's an artist's view of a tumor, and if you took a cross-section where I'm drawing with the mouse here, you could see that you would get a lot of region that's not cellular. And here's another artist's conception, and if you took a cross-section here, you can see the same thing. Sure, you'd come across a few cells, but if you took a cross-section, it might look more like this, or at higher power. Oh, I don't have the higher power, I took that one out. But that the region all the regions that are black are not cells. And so you're missing a lot. In fact, all the stroma gets missed if you're just counting cells that are positive. And so I think it's really important to do molecular compartmentalization as a measurement. And that's why most of the time you do molecular compartmentalization, although there are times where cell segmentation is actually better than molecular compartmentalization, and that's in characterization of lymphocytes or small cells where there's a prominent nucleus, like lymphocytes or other cell types. Segmentation also works really well for cell lines. If you want to image cell lines, because they have no stroma and they're just small cells with a defined large nucleus and small cytoplasm, it generally works well for cell lines, except for stellate cell lines where it fails. So that's what I wanted to talk about, how we measure cells. And now let's talk about high-plex methods. So high-plex methods kind of fall into 3 categories, in my opinion. There's the cycling methods, and there's lots of those. They all suffer from the same advantages and disadvantages, but I'm not going to spend a lot of time talking about cycling methods. Cycling means that you make an image by immunofluorescence or some other method, mostly immunofluorescence. The only exception to that is MCISSS, which is a chromogen-based method, but all the rest are immunofluorescent. And then you wash off or bleach the immunofluorescence and do it again. And you can keep cycling, and sometimes you can cycle as many as 10 or 15 times. And if you have 4 per cycle, you can get up to the 60. And Akoya recently claims to getting to 100 with their cycling method, which is a takeoff of Codex. I think they have a new name for it now, but that's what that is. Then the next high-plex method that's probably more accurate is mass spec IHC, better known as MIBI or imaging mass cytometry. And I'm not going to spend too much time talking about those either, but those can get to sort of 35 to 50 plex. and have a resolution between 1 micron and half a micron. And then the last one, the DNA tag-based HiPlex, is what I'll spend a lot of time talking about. That's the nanostring method. And there are some other DNA tag-based HiPlex methods that I won't talk about today. Just kind of as a summary, I made this kind of summary slide of HiPlex methods of the plex. And you can see digital spatial profiling, or the nanostring method, wins the plex battle for proteins, and also for mRNA as far as I'm concerned. But they're low resolution or non-imaging. They have very 10 to 20 micron resolution. Whereas if you want to have high resolution, you probably want to use imaging mass cytometry, or MIBI, which is the method that was popularized by Gary Nolan and Mike Angelo at Stanford. And the resolution is 10 nanometers for MIBI. So even though it takes forever to get an image, you really get high resolution in the image. So I want to talk about digital spatial profiling, and I don't know if I need to introduce that here, but I will. And the trick of digital spatial profiling is that you take a photocleavable linker, and then you have a unique DNA tag that you can measure the tag, and you conjugate the tag to the antibody. And so it's a HiPlex antibody-based approach, but you have a photocleavable linker, and you determine how much antibody is present on the basis of how much tag you count. And you can count the tag by a barcode that's attached to the tag and hybridizes to it, and then works in a counter, or you can sequence to count the tag, and then just count using next-gen sequencing to count how many of each of these tags there are. And here's how the platform works, is that you define a region of interest, which I'll talk about a little more specifically. You put the antibodies with the tag on, and then you sip up, and then you put a little light on, actually a UV light, to cleave the cleavable tags off, and then you sip up the tags from the defined regions of interest. And that's how digital spatial profiling works. The key slide, and arguably the most important slide, is this one, and that is, how do you define your regions of interest? And one is you can just circle a whole big region and just say that's what we're interested in, that's what we're going to measure. Another is you can be more specific and try to circle individual cells, which is single cells. And the other, which they unfortunately call phenotyping, I call molecular compartmentalization. And that is where you define the region of interest by the expression of a protein. And that's what's done here. So on the right side, you can see the green is all cells expressing cytokeratin, and with some dilation and filling. And the yellow is all expression of macrophages and CD68, and the red is expression of CD45. So if you do, that's molecular compartmentalization, and I think one of the strengths of the nanostring tool. So here's a paper that illustrates that strength. So this is actually the paper where we looked at resistance to trastuzumab in breast cancer, and we found that SMA, or smooth muscle actin, was associated with resistance using digital spatial profiling, only using the DAPI, CD45, and CK compartments. So how is that possible? And that's the point that I want to make, is that, so first let me show you some of the data. So one of the things I like about digital spatial profiling is here we did a 58-plex on 151 patients. So that's a lot of data. And it really gives you the power of discovery, beyond which there are essentially very few other tools that can look at that many patients with that many biomarkers at one time in one experiment. And each of these is the biomarker, as you can see listed on the bottom, and each of them kind of creates their own normal curve, or their own curve of the levels of expression. And this is counts. This is the number of counts, which means essentially the number of antibodies. Their yield is only 2 or 3%, so it's only 2 to 3% of the number of antibodies that are bound in the region of interest that was defined by molecular compartmentalization. And when we did that, we found that, in fact, there were a number of, that SMA overexpression was associated with worse outcome in that is the disease, the counts above a certain level was in the CD45 compartment. So what's going on there? It's because the CD45 compartment actually defines the stroma. But the stroma you would never find with any segmentation, because most of the nuclei for the fibroblasts are out of the plane of section. And if you can't find a nucleus, you can't define a cell with traditional segmentation methods. And so as a result, you can see that we found an interesting result that was probably only discoverable by using molecular compartmentalization. And here's an image of that, and I hope you can see it. It's a little bright in this room for being able to see it. But you can see in this panel especially that the CD45 is the light blue, but because we can't accurately measure only the CD45 compartment, we spill over into the stroma where the SMA is. And that's how we discover that SMA is associated with resistance to trastuzumab. And so, but you shouldn't just take the word for it when you've done something anything done by digital spatially profiling needs to be validated by an orthogonal technique. In my opinion, that should be true of any new technology. If you discover something with a new technology like digital spatial profiling or 10x Visium or whatever, you then need to validate it with something else to make sure it's real. So we validated it with quantitative fluorescence. And so this is measuring alpha SMA in the stromal compartment by DSP on the x-axis, and then the y-axis is measuring alpha SMA in the stromal compartment using a molecular compartmental quantitative immunofluorescence, or AQUA technology. And you can see they correlate pretty well. And in fact, we found the same outcome information also correlated. That is, high SMA was associated with worse outcome, whether we analyzed it by digital spatial profiling or whether we analyzed it by quantitative fluorescence. So next I want to go into some examples of some biomarker discovery. And this is another example of using digital spatial profiling to find markers that are associated with response to immunotherapy. And I showed this slide because this is already published, so that I'm not going to go into too much detail, but you can find the detail in the publication. And specifically, we took a tissue microarray where we made 4 different blocks. And so this is a 4-fold redundant tissue microarray, so we're sampling the heterogeneity, but that also gives us the strength of redundancy. If we find a marker that's present in one and not the other 3, that should raise question, and it does. If it's present in all 4, then it's probably real. And so that's how we've used our higher fold redundancy of TMAs. And here's the sort of the consort diagram. We took 56 cases and 71 targets in 4 compartments, so we had 284 variables. And then we do a univariate and multivariate assessment of association with response to immunotherapy. In this case, we looked for markers associated with overall survival as opposed to progression-free survival. And these are the compartments: cytokeratin, stroma, CD45, and CD68, where stroma is defined as CD45 plus CD68. And then all the ones that are in bold, and I know this is too small to see, but all the ones that are in bold were statistically significant. So that's what we need to validate. We can't just say, okay, voila, we have a new biomarker that predicts response to immunotherapy. We have to now validate that in a second approach. And so that's what we've done. And in fact, that's what you can see here. This is the training set, the initial set of patients that was these, and these are all non-small cell lung cancer patients. And you can see if they have high markers. And in this particular case, we were looking at the CD66 as a biomarker, which is a biomarker in stroma that was statistically significant. We've actually looked at some of these others here, and I'll talk about CD44 in the next presentation, in a couple minutes. But CD66 was an interesting biomarker where we know it's expressed in neutrophils. And so neutrophils are CD45 positive, so we could look in the CD45 positive and see that patients that express neutrophils are associated with resistance to immunotherapy. Don't know why yet. That's the thing about a discovery tool is that it doesn't necessarily tell you why, but it does tell you it's a hypothesis generating technique. And so then we validated it with, this is YTMA 471, which was the discovery cohort. Then we validated it with QIF. So first we validated with quantitative fluorescence on the discovery cohort, and that's shown here. And then we did quantitative immunofluorescence on a completely independent, time-independent cohort. So we collected patients with response to immunotherapy who were collected 2 years later than our first cohort. Each cohort is about 2 years. And then if we want to say that, we can't say it's predictive because it's impossible now to randomize to no immunotherapy. Everybody gets immunotherapy. But to use the term predictive as a biomarker, you have to have a no treatment arm. And since we didn't have a no treatment arm, we could look at pre-immunotherapy treatment and prove at least it's not prognostic. And that's what this shows, is that we're not prognostic because CD66, whether it's high or low, makes no difference in a patient cohort when they're not treated with immunotherapy, which is what's shown over here. So this is an example of discovery of a biomarker that we wouldn't really had a basic science hypothesis reason, but it's toward our ultimate goal of finding biomarkers of patients who are resistant to immunotherapy. Since the drug companies will find plenty of patients who will respond to immunotherapy and they want to make biomarkers to predict response, I think as immunotherapy moves to the adjuvant setting, we'll want to find out patients who should not get drug because of the risks of immunotherapy. And this is the first step toward that, identifying CD66. We're now following this up with secondary and potentially, hopefully in the future, prospective studies. This is another example of a similar biomarker discovered in the same cohort. And I'll go a little faster. Again, we had all again, it was done in a tissue microarray, in this case, twofold redundant tissue microarray, and then validated with a collaborator who was a former fellow in my lab from the 12th of October Hospital in Madrid. And you can see again, we did the molecular compartmentalization, and again, we show the number of the signal-to-noise ratio, but also the number of different variables. In this case, was 77 different markers on about 150 patients. And then we do the same thing where we did a multivariate analysis and looked in the various compartments, and then looked at the cut points for association with response to therapy. And there are a number of candidates that we then had to validate. So then to validate, you go back to the training cohort, but validate them with quantitative immunofluorescence, because just trusting DSP, I think, was a little bit of a stretch. This DSP is a great discovery tool, but I always believe that you need to have an orthogonal confirmation of that result. And that's what, these are just some representative QIF images. And here we found that CD44 discovered by DSP now validates by quantitative immunofluorescence, whereas in this case, it was the reverse, that high CD44 does better. So in looking for markers for resistance, this isn't a great marker because it's loss of CD44 that's associated with resistance to immunotherapy. And loss of a marker is never as good as expression of a marker if you want to make eventually you want to make a diagnostic test that shows resistance to immunotherapy. But this is kind of where we are. Just another example. This is also now published as a biomarker discovered by DSP. Again, we don't have the biology behind it. This is not basic science. This is reverse translational approach to try to find biomarkers associated with resistance to immunotherapy. So I'm going to continue on that theme, and now we're actually doing pretty well time-wise. So I'll now talk about doing the whole transcriptome assay. I know that some of you are interested in the whole transcriptome assay. You have a question?
Yeah, just before you go into that, you had multiple hits that were statistically associated, and you said you validated all of them. So did CD44 pop up as the only one that's validated, or are there multiple things?
So CD66 validated as well, but some of them didn't validate. So like Vista, EpChem didn't validate.
Meaning that when you stain,
That is that when we did immunofluorescence staining, it didn't mimic what the DSP told us. It didn't confirm the DSP result. So when I get reviews of paper, when I'm asked to review papers that just show me DSP, I always reject them. You need to have a validation technique. DSP is a discovery tool, and you can use it as a validation tool too, but then you need a second independent cohort. And if you only have one cohort, then you need to validate with, in my opinion, you need to validate with an orthogonal technology. And you can use immunofluorescence, you can use IHC, whatever you want to do, but you need to have something beyond DSP, because DSP is subject to high levels of false discovery, as you could see here. And it depends on your cut point as well. If I go back a couple slides, you can see these are the univariate variables and the p-values. And so there's lots of markers here like phospho J and K that has a p-value that's just barely significant, but it's not significant in the multivariate analysis, so we didn't bother validating that one. Whereas things that were significant in the univariate analysis, like CD66, was significant in both the univariate and multivariate analysis, so we started with validating that one with QIF, and it turned out to validate, and that resulted in this data and ultimately that publication, which I showed, this publication. So now let's switch our hats to the mRNA, and this is, I think, in some ways, the exciting and the reason to come give grand rounds at UWash or any place that it gives a grand rounds is I can talk about stuff that's not published, which is a lot more interesting than the published stuff to you potentially. And but apologies in advance, I'm not going to show you any of the genes. I'm just going to put XXXX because we can't talk about it if we might potentially patent it. And I'm frequently reminded that Yale owns my work and they will make the decision on what we patent and what we don't. And so I'm not disclosing anything that I shouldn't disclose, but don't be offended by the fact that I'm not showing you the genes quite yet, because that's the advantage of a seminar. I can show you stuff that's not published yet, even though to this point I've shown you mostly published work. So this is the cool thing about DSP is you can do RNA as well as protein. But with RNA, theoretically, you could do 18,000 plex. And it's all based on in situ hybridization using the Z-probe method that is popular with RNAscope or was invented by RNAscope. And so it's a pretty robust method for in situ hybridization. It has a very low false positive rate. And then it's the same way. You define the regions of interest. You can do molecular definitions, or you can do a manual definition of the region of interest. Then you go back and zip them up, and then you sequence them. And in this case, you need to sequence the barcodes when you're doing, or you need to sequence the DNA tags because there aren't enough barcodes to get to 18,000 plex. But by sequencing, you can easily do 18,000 plex. But then what's the pipeline? And so when I say, what is the pipeline? Here's an example of the pipeline for RNA-seq. And this was developed over many years, and people kind of came up with how you start with the FASTA files or the FASTQ files, and then what you need to, how you deal with that data, and how you ultimately can figure out how many RNAs there are. But there's quite a bit of computational work that needs to be done in there. And this computational pipeline, which is now mostly open source and fairly commonly used for RNA-seq, doesn't exist for DSP. So what do we have to do? So we had to invent our own pipeline. And so we invented 3 different pipelines, which I'll show you about. And if you all are doing DSP with RNA, I know some of you are, even though you're not in the room, that I would invite you to try to use some of these pipelines as a way to go. And our pipelines are the redundancy pipeline, which I already gave you a hint about, the delta pipeline in the one cohort where you have both the pre and the post specimens. So the delta is how did it change with therapy, and does that therapy if the levels of various messenger RNAs or various genes changed, then presumably they must be important to the therapy. And then the train test validate, which is a more conventional approach to a pipeline. Here's the redundancy pipeline, and this is tissue from 56 different patients. But again, this was 4 different blocks, and this is kind of a schematic of where those cores came from. They made 4 TMA blocks, and the idea is that in each of these, we got somewhere between 130 and 150 discoveries of RNA genes that were associated with resistance to immunotherapy or response to immunotherapy. But if you looked at only the ones that are in all 4, you get a filter that isn't available using any other technique. And so this is sort of a redundancy filter, and you can see that, I think you can see that here, that these are the genes that come out. There was, in the CK high expression compartment, that is the CK molecular compartment, there were only 12 genes that were associated with shorter overall survival, or resistance to immunotherapy, and is another way of saying that. And there were only 12 genes out of 18,000 that were present in all 4 blocks. And so one way to look at that was with the deconvolution analysis. And this doesn't help me that much. It makes a pretty picture, and you can see sort of the distribution of different genes, and I'm not even going to explain it, but this is what comes with the software. And this is what comes with the NanoString. This is how if you use NanoString approaches to analyze your data, these are the data plots that you get, which it didn't help me that much. So what we did is do what we'd done before, is we looked at all 4 blocks and then looked at the log rank p-values of overall survival and the hazard ratio. So you can't compare p-values. If you're a biostatistician, they'll tell you, but you can compare hazard ratios. So we looked at the hazard ratios of all 4 blocks, and then at markers that were present in all 4 blocks. And these 2 markers came up as being present and significant in all 4 blocks. And so that's why these 2 markers are potentially interesting genes. And would it be possible that we could create a 2-gene signature in the CK compartment that is actually associated with resistance to immunotherapy. And that's what we have. This is in the CK compartment, the cytokeratin compartment. And you can see that in overall survival, it doesn't quite make it. In progression-free survival, it's highly statistically significant for associated with worse outcome. And this is the training set, and this is the validation set. So these are two completely unique cohorts of non-small cell lung cancer patients. And so we're optimistic that this 2-gene signature may be a gene signature that we can take now and prospectively test for resistance to immunotherapy in non-small cell lung cancer. We're not going to use the DSP to prospectively test it. We're going to do it with RNAscope and just do it with sort of conventional RNAscope in situ measurement with quantitative fluorescence. But we wouldn't have found this without the high-plex use of the digital spatial profiling. And then we can look in TCGA and look at these 2 genes, and in fact, they're not prognostic. So they're tightly associated with response to therapy, but have no prognostic value when you combine them. And that's what you see here is the lack of prognostic value and lack of survival. So we're guardedly optimistic that this 2-gene signature might be associated with response to immunotherapy. Now that was in lung cancer. The next one I want to tell you about is in head and neck carcinoma. And in this one, working with my colleagues in Athens, Greece, Amanda Siri and Nikki Gaviolatu, they did a trial like this where the trial was led by Amanda Siri, and they took a preoperative specimen, then they treated with nivolumab, and then they took a postoperative specimen, and in some cases they even have a third specimen from some patients. So the idea behind this filter is that if you take the pre and the post and something changes between pre and post, which is shown here, all the things that the red is increased in post-treatment and the blue is increased in pre-treatment, that those changes those genes might be important in resistance to immunotherapy since they're changed by immunotherapy. And so that gene list is shown here, not completely, but the AUCs for response to immunotherapy are shown here. And the best we did in this pre-post pipeline was an area under the curve of 0.72 in the cytokeratin compartment, and that one required 7 genes. And then with 6 genes, we could get to a 0.83 in the CD45 compartment or 0.68 in the CD68 compartment. And this one we haven't validated yet. We're still in the process, so I feel less guilty about showing you XXX because it might all be wrong. We need to validate on a second cohort, and I've just hired a new postdoc that will try to do that validation. But one thing that makes me think it's probably right is that in the CD45 specific signature, this 6-gene signature for CD45 that had an area under the curve of 0.83, when we checked other compartments like the CD68 compartment or the CK compartment or the stromal compartment, which is CD68 plus CD44, the area under the curves all became awful, or they all went down. So that suggests to me that the area under the curve is specific to the spatial information that in which we're collecting the RNA or the genes of interest. So the last one is the train test validate using Lasso, which is kind of more of a traditional pipeline with a training set and a validation set. In this case, we have I'm just going to show the training set data where we had 80% of the cohort was split up. We took our cohort of about 60 patients and split it 80-20, and then we looked for genes that were different and then did a Lasso and built the models and then validated on the 20%. And this is much more typical to how models are built with mRNA for bulk mRNA. This is how people build signature models. And our signature models were then built to look specifically in each compartment. And much to our surprise, instead of having models in the 0.7 range, which is what you can always get with a training set, or 0.8, we had areas under the curve as high as 0.95, 0.97. This 8-gene CD68 marker has an area under the curve of point. Now this is the discovery set, so don't get too excited. And I had to tell my postdoc that. I said, this is the discovery set, so of course you're going to overfit. But then she showed me this slide, and I said, wow, that is pretty cool. Because if you take those gene sets and put them in the wrong compartment, they all become no good anymore. So that tells you the importance of the spatial information collection of the compartment. And in fact, what we found that the spatial information generated by CyberSort, the way to specifically tell different genes apart, doesn't work nearly as well as the spatial information obtained by digital spatial profiling. So these gene signatures that in this CD45 gene signature that had a really high area under the curve, this gene signature, actually it's this one, has an area under the curve of 0.97 in the CD68 compartment. We've now validated this by doing DSP on a completely independent cohort of lung cancer patients, and the area under the curve is 0.85 in the validation set. So this 8-gene signature, I think, has legs, and it's also interesting that it's specific to the CD68 compartment. It doesn't work in bulk RNA, it doesn't work in the CD45 compartment, and it doesn't work in this case it's a melanoma, so it's an S100B compartment. And that's what these show. This is very preliminary data, and you can spend more time on it, but I want to move a little more quickly so I can spend the last 10 minutes talking about the next topic, and that is bringing things to a CLIA lab. And if there's time, we can do questions at the end. So will HiPlex ever come to a CLIA lab? And my answer to that question, I think, is no. During my career, the distressing truth is that in the research world, we've got new HiPlex methods every year. We've become more quantitative. We're faster with data collection. We have more computational power, more powerful imaging software. In the meantime, what's going on in our CLIA lab? We're getting worse. The companion diagnostics are systematically worse. PD-L1 is the worst companion diagnostic ever invented. It's not accurate and it's not tightly associated with response to therapy, but it is FDA approved, and our oncologists insist on it. This happened now with Ki-67, which was approved at a non-reproducible cut point for abemaciclib in breast cancer. So there's more scoring systems with less reproducibility in the clinic, and drug analyte assays using the same analyte are getting worse, and the new amplification systems are moving the clinic more and more toward binary results, which in some sense is good. If you only have 2 choices, your chances of guessing right are better than if you have 5 choices. And the concordance amongst pathologists is awful when they get 5 choices, but when they get only 2, can be pretty good. And so I think that what's going on in the clinical world is actually going in the opposite direction. And unfortunately, I don't think any of the benefits of the HiPlex world are making their way to the clinic. So the last thing I want to talk to you about in the last 5 minutes or 10 minutes or so is trying to bring some quantitative result to the CLIA world. And that's in the context of these new drugs called ADCs. And since I'm speaking to a pretty sophisticated audience, I'm going to assume that you all know what ADCs are. But this is the poster child example, trastuzumab deruxtecan. And this is where they attach toxins to an antibody, in this case trastuzumab, and the toxin deruxtecan is an inhibitor of topoisomerase. And the advantage of this is that when it gets taken up and then the linker gets cleaved off in the cell, it not only kills the cell, but it leaks out to the adjacent cell and has what's called a proximity or neighborhood effect, and can kill other cells as well. And for that reason, this drug did extremely well. And I don't have my full slide set in here to give a talk, an hour talk on this like I will give tomorrow at Raresight, but rather I want to just show you a little bit of this. And this was what attracted a lot of attention, and is that it worked not only in the 3+ patients, but in the 1+ and 2+. And these are waterfall plots showing how well it worked. And in fact, this is from the DESTINY-6 trial, or DESTINY-4 trial, which was announced at ASCO. And in fact, this is the ASCO, the slide from the New England Journal paper published simultaneously with the presentation at ASCO showing a doubling in median progression-free survival in advanced stage breast cancer by giving trastuzumab deruxtecan to patients without amplified HER2, in unamplified or low levels of HER2, but not 0. They had to be +1 or +2 in this study. And so this suggested that these ADCs are pretty powerful, but that we need a companion diagnostic test to pick out the patients who express HER2. And that's what the excitement is about. I will say this drug was the first time that a standing ovation was given at ASCO to the presentation since trastuzumab, which was 25 years ago. So I think that this drug will likely impact care a lot, and it falls to the pathologist to pick the right patients. And they have to be 0s, not 1s. they gave the drug to zeros, and this was a parallel trial called the DAISY trial run by Fabrice André in France. And what they found is that some of the zeros, and these are the zeros here, I think in cohort 3, some of the zeros actually benefit. So does that mean they were actually zeros, or were there plus ones that were misassigned as zeros? And we're trying to figure that out. But what's happened is that this now changes the equation for breast cancer management. It used to be that the zeros and ones were negative and the 3 pluses were positive. And if it was 2, we could reflex to FISH and figure out whether or not the patient should get drug. And it worked pretty well. But now, we need to, the cut point has gone from 2 to 0. So how do we adjudicate the IHC equals zeros? How do we know which of the IHC zeros will benefit and which will not benefit? And this paper that we published in JAMA Oncology suggests that pathologists are pretty good at telling 2s from 3s, and we have an adjudicating test, FISH. But we're really bad at telling zeros from 1s. And that's what's shown here. These are the percentage of pathologists that called 0 versus the percentage of pathologists called 1, 2, 3. And you can see almost half the cases were not agreed on by 18 different pathologists. And so this is, I think, the best reason for this is that we're using a scale in the wrong dynamic range. And this was in a recent review that just came out that sort of indicts the FDA for approving this. That came out in a European journal. The European pathologist said it's like trying to weigh mice on scales built for elephants, which is a really great analogy because it's in the wrong dynamic range. And we actually knew this since 2005. This is some work from my lab in 2005 where we realized that there's 2 different kinds of HER2 expressed in breast cancers. There's the amplified cases, and the amplified cases can be separated from the unamplified cases with just a breath of antibody. You don't need much antibody, and this is how the current IHC test is, and 70% of the world uses 4B5 in their IHC lab to tell the amplified cases from the unamplified cases, and it works fine. But what if you want to tell apart the low cases? Well, then you need to add a lot more antibody. But you can then tell apart the low cases, but the high cases are all in the saturated range and are not useful. And so we looked at this and said, well, we should fix that problem. We can make a high sensitivity HER2 assay that can then be the adjudicator for IHC equals 0. And that's what we did, and that's what I wanted to show a little bit because we've had our first 2 tests ordered this week. So this is the way we did it is we took a bunch of cell lines that have low level expression of HER2, and then we quantified them by mass spec, and then we put them into an array in opposite orientation that we can run. And the way we quantified them is we grew up a bunch of cell lines and then used QPath to determine the area of cells and mass spec to determine the attimoles. So then we could determine the attimoles per square millimeter for each cell. And then we can use this cell line to make a standard curve that looks like this. Throw away the amplifieds. That's not interesting. We already have assays for amplified HER2. We need assays for non-amplified HER2. And to make a high sensitivity assay, and look at this nice linear standard curve, just like a Bradford assay or a Lauer assay from your college biochemistry, we can now use a standard curve to determine how many attimoles per square millimeter there are as a function of our response indication when we scan either by quantitative fluorescence or by other methods. And if you think back to what makes a good assay, well, for a good assay, you need to have a broad linear range. Every assay saturates eventually. You want the broadest linear range with the lowest possible limit of quantification or limit of detection. And that's what we're going for, and the highest possible limit of linearity. And that's what you can see. If we use a standard assay, the standard amount of HER2, the current 4B5 assay, you get this. All the low cell lines are basically negative, and the high cell lines are positive. But if you use our high sensitivity assay, or just put more antibody in, basically, then you can stratify the low-level HER2 expressors, and the highs are saturated, which is fine. So this is how we built our high sensitivity HER2 assay. And if you look at it, here's an example of a +1 case. And the +1 case shows there is actually plenty of HER2 there, plenty of target there. No surprise, trastuzumab deruxtecan is going to work on these patients. They have a fair bit of HER2 expression. So then we asked the question, how many of these are in a real population of breast cancer patients? So we retrospectively collected 350 plus cases from 2010 to 2014 at Yale, made a tissue microarray around them, and quantified the tissue microarray. And that's what's shown here. The zeros are blue, the ones are red, the twos are black, and the threes are green. And you can see most of the greens are up here. and where you'd expect them to be. But look how many blues, the zeros, are present above the limit of detection. So here's our limit of quantification here. And there's lots of blues in this middle range, suggesting that there will be lots of patients who were called zero who are going to benefit, which is exactly what they saw in the DAISY trial. And so why are they leaving these patients on the table? Why are we using a pathologist-based assay reading HER2 when we should be measuring? And if we measured, wouldn't we get a lot more patients benefiting from this drug? So I'm trying to convince AstraZeneca about that. And they're going to apparently let us do a prospective trial on this, but we can't retrospectively access their DAISY-4 or the DESTINY-4 or DESTINY-6 trial patients. So I'm going to sort of skip the summary, but the idea of the summary is that there may be probably as many as 70 or 80% of the cases that express some HER2, maybe as many as 90% above the limit of detection, and that a quantitative HER2 assay could be envisioned as a reflex test for a HER2 zero. So you don't just have to call them all ones, which I'm afraid is what's happening, is that pathologists are being bypassed. The oncologist, when you call a zero, you say, are you sure it's zero? You know, I can't give the right drug if you call it a zero, and I can help the patient if you call it a one. Okay, I'll call it a one. If that's the way we do pathologists this day, pathologists are not long for this world. I really think that if the pathologist meaning is a political position, not a biochemical position or a biochemical, if we're not using our professional expertise, but rather just being convinced for political reasons, no reason to do the assay. So how do we validate that? So if you validate in an IHC lab, and unfortunately Maria's left, but she runs your IHC lab, she has to go through the Fitzgibbons et al. Principles of validation. But that's for validating read IHC, not measured IHC. So we made up our own validation system for high sensitivity, the high sensitivity assay, since it's an analytic assay. And we borrowed the terms and the things that we needed to do from analytic chemistry assays. And we're running out of time. We're actually, I think, 2 minutes away, so I'm going to cruise through this and not show this whole part. Suffice it to say that we validated it using the FDA's guidance to industry. We found the limit of detection, limit of quantification, and now we tested the accuracy and the precision of the assay, and then we did clinical validation on actual core biopsies, not just cell lines, so that we knew that they were actually true. It's actually about 90 to 92% of the patients are above the limit of detection for HER2. So I think there's a lot of patients that are going to benefit. But there's 8% of the patients, or maybe 10%, that really don't express any HER2, and 10% of those who get, if they gave them trastuzumab deruxtecan, would get interstitial lung disease that can sometimes be fatal. And so this drug shouldn't be given out like trastuzumab, which basically doesn't kill people, since this drug has some major toxicities. And this is just, we're kind of running out of time, so I'm not going to show all the rest of this. But I do need to, this is how we're going to report it. We got our first two. This is our workflow with our current system. And then one of the reasons I'm in Seattle is to meet with this company, Raresight, which we're porting over and doing a bridging study to the Raresight platform, which will allow us to do much more high throughput. A scan on our old platform, a scan on this platform takes 4 hours. A scan of that same core biopsy on this platform takes 4 minutes. So our throughput will be much improved once we get to this platform. And with that, I just want to show the people in my lab who actually did the work, and then the lab group last year, and then this year, not as many people could make it, and my lab's downsizing a little bit. Then I think I need to finish with this slide that you can all sign in and evaluate or take a picture with your phone and give me good ratings, because I'm the first grand rounds of the year. Thanks very much. Are there any questions, or should we bolt? Because,
No, until people come into the room.
okay, well, why don't we take questions until we get booted out? Go ahead.
It seems like a fair number of the new biomarkers are dependent on molecular compartmentalization. So I assume that you also are incorporating that into the validation studies, and that would also be a...
Right, so molecular compartmentalization has an unfortunate history. We invented molecular compartmentalization in 2001 at Yale, and Yale patented it. And we founded a company, and I was guilty as charged. We founded a company called HistoRx in 2005, and Yale gave them the exclusive license. And the company was bought in 2012 by Novartis Genoptics, which now owns the exclusive license to molecular compartmentalization. And that's why lots of other people haven't done it. And that was not what we had in mind when we invented molecular compartmentalization. We wanted it to be broadly used. In the interim, software has all become open source. Now there's open source software called QuPath, which is an open source image analysis software. And so even though I didn't include it in this presentation and won't till for another year or so probably, we've now invented Aqua on QuPath. So now you can do molecular compartmentalization on open source software. And in a month or two, or a few months, we'll tell you the GitHub site where you can download your own ability to do this without having to worry about the patent and the license that prevented molecular compartmentalization from becoming a popular measurement technique.
Dave, can you comment on whether there's a problem with the part of the tumor that's biopsied and how you...
So that's the problem of heterogeneity is a huge problem because it's the standard of care for breast cancer to do one core biopsy or maybe 2 or 3 of the same location. In prostate, I know they do more core biopsies, but oftentimes only one of the core biopsies will show prostate cancer. And that's just what we have to do. We can't take out the whole tumor and biopsy and measure the whole tumor. That's just not the practice of medicine, and I don't think will ever be the standard of medicine. But I say if you can find a biomarker that works on a very small sample, it will work on a big sample. The reverse is not true. If you find a biomarker that only works on one region, only one of the 4 TMA spots, for example, that's not a very good biomarker because it's subject to heterogeneity. So when you're doing your biomarker discovery, you want to do biomarker discovery with redundancy from multiple regions of a tumor so that your biomarker is robust to heterogeneity.
I have 2 questions. So the first one is, completely agree that I think the FDA approval of treatment in HER2 low group was premature, and that we don't have the right assay for it, and that the recommendation kind of does a little bit of a bias as a pathologist. So until we have a quantitative assay, the one that you've, similar to what you developed and can employ, how do you recommend handle those situations where we're asked to look at HER2 low?
So I think you should do the best you can. You might try to convince so the array that I showed, this array is commercially available. You can buy this from Array Science. So you can make your own assay if you're particularly industrious that has the right dynamic range for HER2 low. You could have your own HER2 low and your own HER2 high assay just by using this standard with every run. And it's not very expensive. You can buy a block of this for like $2,000 or $3,000. And in one block of this 14-spot array, you get 300 assays. So that ought to keep you busy for a while. So that's one way around this problem. The other way is just to kind of be rolled over, which is what the FDA would have us do, and just call them all +1. And maybe if you have enough patients with metastatic breast cancer, one will die because they get interstitial lung disease. But that's not on you. That's on the drug company. But I don't have a good answer. How do you weigh? Asking me, so how do you weigh mice on a scale built for elephants? I can make something up. That's what David Hicks is going to try to do in the CAP. All of the pathologists in the room probably got the advertisement for David Hicks speaking on behalf of AstraZeneca, Daiichi Sankyo, on how to read HER2 low. Teaching us how to weigh mice on a scale for elephants is not really legitimate, in my opinion. You need to make the scale correct before you expect us to do it. And it's really taking advantage. It's making pathologists look really stupid. And so my hackles come up when I see those kind of advertisements. And even though David Hicks and I are good friends, it bothers me that he's willing to sort of sell himself to pharma to sort of forward their agenda. And he's a highly respected pathologist. Well, if David Hicks can do it, I should be able to do it. Then that means somebody can do it. So I'll learn from David Hicks and I'll figure out how to do it. Unfortunately, that's what's happened with PD-L1, that, they claimed they could do it, and the experts could tell it's SP142. It's just, you just needed more training. Well, that turned out not to be true. When they did the clinical trial again, it failed. So it wasn't about training, it was about a bad assay. And I think that's what's going to happen with Ki-67 is if we don't go quantitative, the abemaciclib trials will probably not pan out. And similarly, I think for HER2, I know that both Dako and Ventana are working on a HER2 low assay. So that's what will save us as pathologists, is we'll switch over and we'll do 2 HER2 assays. We'll do high HER2 for regular breast cancer, and then when we get a metastatic cancer, our oncology colleagues will tell us this is a specimen that we might give and HER2 to those patients, or trastuzumab deruxtecan, and so then we'll do a different assay for them. And poor Maria will have to validate 2 HER2 assays in her lab, but that's not that big a deal.
Second question, I recognize some of these don't have, great answers. I'm just curious your opinion on some of this. So, thinking back to, when you're looking at the research, the ability to do all this plexing in the research and then what we're actually doing in the clinical field lab. I think about that all the time because we're always trying to control for pre-analytical variables, and I'm just curious, like in the Yale system and systems you've seen, it seems like criteria for increasing controlling for quantitative or pre-analytical variables like cholestemia time, formalin optimization time, etc. It always sounds great on paper, like, hey, just do this, get it in formalin within an hour, make this done within an hour, etc. But then you hit the real world, right? The surgeon's doing this and that, and I mean, everything becomes, it's not as clear cut and as easy as one would think. And I actually think it's much easier in smaller systems, like maybe small private groups. When you get large academic multi-hospital systems where specimens are being transferred to a central lab and all those sorts of other things, it gets really hard to do. And so, this is from someone who has a research lab specifically in biomarkers. I mean, all of your studies are so carefully controlled, and then there's life.
So then you have to, so that's a great comment, because I believe that ultimately you have to discover with whatever platform you can discover with, and then you have to validate on core biopsies. Core biopsies are the only specimen which have controlled pre-analytic conditions. You take the core, you put it in formalin, and if your interventional radiologists or surgeons are not doing that, instruct them to do that. And you'll know if they're not doing it, because every once in a while you'll see a dried out core. So when you see a dried out core from someone, that's a huge red flag. Go talk to that person. Say you must be putting him on a Telfa pad for too long. Right into formalin. And then you get rid of your pre-analytic variables. And that's anything you discover using DSP or any platform. If you can't validate it on core biopsies, it's not worth, it'll never make it to the clinic.
And so we do that. The challenge for us is incidental. So if someone's breast cancer, or they're getting a bilateral mastectomy for BRCA and they have breast cancer.
I'm sorry.
Incidental findings. So someone who's getting a bilateral mastectomy for BRCA, and then it just so happens that
Have a core biopsy first.
Well, there's no lesion.
Oh, there's no lesion.
Yeah, just prophylactic.
And then you find something. Fortunately, those are relatively rare.
It's uncommon.
Yeah, and so in those uncommon cases, you just have to deal with the fact that that sat on the bench for 3 hours, even though the tech marked 1 hour.
Yes.
Great.
Thank you again.
Well, thank you all, and thanks for coming, and those who are online, thanks for listening.